My name is Tiwari, and today I am going to discuss a few topics of Biology Class 12, Chapter number 2, that is called Sexual Reproduction in Flowering Plants. So friends, before I start this chapter, I'd like to tell you all that this content has been developed by me for the welfare of student community so that they could learn at the comfort of their homes. But at the same time, I would like to remind you all that this content is purely for educational purpose. It should not be used for any commercial purpose. As far as the references are concerned, you all know that NCERT textbook is the best reference book for class 12 biology. I have images I have taken mostly from the NCERT textbook only, but at few places, in order to make this presentation more interesting and more interactive, I have also taken the images from the website google.com. For the video lecture, I generally prefer the online labs, which gives you a wonderful opportunity to learn the activities in biology of this chapter. So friends, let's discuss about the learning objectives of this chapter. After reading this chapter, you will learn that flower is a fascinating organ of angiosperms. Then you will also learn the definition of a flower from the biologist point of view. Then we will discuss the structure of stamen, then microsporangium and pollen grains. Then we will also discuss in details about the female sex organ, that is pistil, the megasporangium, that is ovule, and the embryo sac, that is also called as female gametophyte. So friends, we know that flower is a fascinating organ of angiosperms. Flowers, I don't know, from time immemorial, I should say, I don't know when the various use of flowers started, but now we know that the flowers are widely used as objects of aesthetic, ornamental, social, religious and cultural value. They are used as symbols for conveying the important human feelings like love, affection, happiness, grief, mourning. If you want to wish your friend happy birthday, you can wish happy birthday. You can express your sorrow by offering flowers. You can decorate your god and goddesses. You can just say, hi, good morning. Or in the marriage ceremony, you can decorate your place like this. So, what is a flower? So far as the biologist point of view is concerned, flower is a morphological and embryological marvel and the sites of sexual reproduction. These are those structures which are involved in the process of sexual reproduction. So how do you define a flower? Flower is a modified shoot meant for the process of sexual reproduction in plants. Okay students, now let's have a rapid look of the structure of a typical flower. From here you can see that at the base this is the stalk of the flower. Okay, this is the stalk of the flower. Then the outermost part of the flower is called calyx. This is the outermost part. Mostly it is green in color. This is calyx, and one unit of this calyx is called as sepal. The second important part of the flower is the corolla. This is the second part of the flower, that is corolla. The bright, variously colored and most attractive flower part of a flower called corolla. And you know that one unit of corolla is also called as petal. Then these are the androecium or male sex organ. Androecium or the male sex organ. Here also, here and here. Androecium, that is male sex organ. And the gynoecium which is female sex organ. Stigma, then long tubular structure style, and then flask separated structure called ovary. And all these floral walls, that is calyx, corolla, androecium and gynoecium, are present over a flat structure that is called as thalamus. Okay, let me take the help of another flower. The flower, it bears reproductive organs. Here you can see the female sex organ clearly, that is stigma, the long tubular style and the ovary. Together it is called as pistil. This is the pistil. The anther and the filament, they are together called as stamens. Students, in this chapter we will discuss about the androecium. One unit of the androecium is called stamen, and the female sex organ, that is called gynoecium, and one unit of gynoecium, that is female reproductive unit, that is called carpel or pistil. Now it's time to discuss in detail about the structure of the male sex organ, that is called stamen. So from the diagram you can see that the anther consists of, the stamen consists of anther and filament. Anther and filament. Now from this diagram you can see that the anther consists of how many lobes? The anther consists of two lobes. This is one lobe and this is another lobe. That means two parts. But when you cut open, when you make a transverse section, what is the meaning of a transverse section? Let me explain you the meaning of transverse section. When we take the blade and cut anything in the vertical manner, that is from top to bottom, this is called vertical section. And when we cut any object in a horizontal manner, this is called as the transverse section or horizontal section. So what has been done? This anther has been cut into thin slice in a transverse manner. So the transverse section of a anther, it clearly shows that this is one lobe. Okay, this is one lobe and this is the another lobe, and these two lobes are held together at one place called connective. Here this is the cellular diagram, the structure which is visible in a microscope. We can say that this is one lobule or one lobe of anther, and this is the another lobe of anther. Okay, so these are the two lobes. So we say that anther is bilobed. What is anther is called as? Anther is popularly called as bilobed structure. This is called dithecous. That means the term used to explain the bilobed condition is dithecous. Is it clear? Then these two lobes are joined together at this point. Okay, is it clear? So this point is called as connective. This point is called as connective. So two lobes are held together by the help of a tissue called connective. Okay, now here when you observe the one lobe, what you will find? You will find that each lobe has two sporangium. This is the spherical structure. This is the first sporangium and this is the second sporangium. Okay, so each lobe has how many sporangium? Two sporangium. The second lobe also has these two sporangium. Okay, so in this way we find that in an anther, how many sporangium you are finding? You are finding 4 sporangium. 4 sporangia means this is the tetrasporangiate condition. This is called tetrasporangiate condition. Once again, let me tell you, the question is asked in the board exam that why anther is called as dithecous and tetrasporangiate. Why it is called dithecous? Because anther has how many lobes? 2 lobes. Each anther has how many lobes? 2 lobes. And why it is called tetrasporangiate? Because a total anther has how many sporangium? 4 sporangium. So it is called as tetrasporangiate. Now friends, this is the time to discuss in detail about the internal structure of an anther. So in the previous slide we saw that this is one sporangium. When this is called line diagram of the anther, transverse section of an anther. This structure which I am showing with the help of the laser pointer, this is the transverse section of an anther in which cells have not been drawn. Only the details has been discussed here by the help of drawing the lines. So this is called as line diagram. If you are asked to draw a line diagram showing all the details, you will draw this diagram. Okay, and if you have to show the cellular diagram, you will draw this diagram. So what are the structural details? If you will try to find out from outside inwards, it has clearly the 4 layers. Let me use the highlighter. This is the outermost layer. This is called epidermis. This is called epidermis. And the second layer is called endothecium. Just below the epidermis is the endothecium. You can see the arrow. So the third layer consists of 2 layers. The third wall layer of the sporangium consists of 2 layers. They are called as middle layers. So this is the first middle layer and the second middle layers. You can see the 2 middle layers. Okay, and then the innermost layer is called tapetum. In this way, what we find? We find that each microsporangium, okay, each microsporangium consists of the first layer, that is called epidermis. The second layer is called endothecium. The third layer, which is two layered middle layers, 2 middle layers. So these are the first 3 from outside inwards: epidermis, endothecium, middle layers. They help in the protection and dehiscence of the anther. What is the meaning of dehiscence? Dehiscence means when the anther it becomes dry, it splits open and releases the pollen grains which are formed inside the microsporangium. Okay, so the first 3 layers is clear: epidermis, endothecium and middle layers. Now the innermost layer, that is the 4th layer, This is very, very important. This is important in the sense that it provides nourishment to the developing pollen grains. Okay. So what is the characteristic feature of tapetum? It has dense cytoplasm and it has more than one nucleus. Many a times you will have questions like, what is tapetum? What are the characteristic features of the cells of tapetum and what is its importance? What are the characteristic features of tapetum? Number one, it has dense cytoplasm. Okay, it is the innermost layer of the microsporangium. It has dense cytoplasm and it has more than one nucleus. What are the functions of tapetum? Tapetum helps in nourishing or providing nourishment to the developing pollen grains. So here this microsporangium has 4 wall layers and the inside layer is full of sporogenous tissue. They will develop into microspores or pollen grains. So what is the process of microsporogenesis? What is microsporogenesis? Microsporogenesis is the process of formation of microspores to form a pollen from a pollen mother cell through meiosis. In the previous slide, let me once again go back to previous slide. Students, you find that these were the 4 wall layers, okay, and then this layer is called tapetum. This layer which I am highlighting is tapetum, okay. This is the tapetum. Now you can see I have shown all these wall layers with yellow color, okay. Now what about this? Let me use the laser pointer. What about this? This cell also are large in size and they have prominent nucleus. You can see the prominent large spherical nucleus present in this. This central portion of the microsporangium is called sporogenous tissue, where you will find microspore mother cells. Microspore mother cells are diploid. They will undergo meiotic division. Okay, let me move again to the next slide. So what I was telling that what is microsporogenesis? It is the process of formation of microspores from a pollen mother cell through meiosis. So this is the microspore mother cell. This is one microspore mother cell, or it is also called as pollen mother cell. Okay, so what will be the chromosome number? What will be the ploidy level of microspore mother cell? It will be 2n. What will be its ploidy level? It will be 2n. Okay, let me use pen here. It will be 2n. So when meiosis 1 will take place, one microspore mother cell will divide into 2 haploid, okay, microspore dyad. That means 2 cells. Now as a result of meiosis 2, second meiotic division, you will have 4 microspores. 4 microspores, that means microspore tetrad. Initially, this is the first microspore, this is the second, this is the third, and this is the fourth microspore. So altogether, as a result of meiosis 1 and meiosis 2, in the microspore mother cell, how many microspores are formed ultimately? 4 microspores. Okay, this process is called as microsporogenesis. Since this all these 4 microspores in the beginning, as soon as they are formed, are lying attached to each other. They are lying attached to each other, and that is why they are called as microspore tetrad. All the 4 microspores are together. So by the help of this diagram, this is the diagram which has been given in your NCERT textbook. These are the 4 microspores held together. Now when after some time they will get separated from each other. So this is one such microspore. Okay. Now what will happen? This microspore will gradually develop into a pollen grain. Sometimes students, we use the term microspore and pollen grain as synonymous terms, but there is little difference. The microspore is the structure which is formed as a result of meiosis, and all the 4 microspores are held together in the form of a microspore tetrad. But when they are separate, they will occupy somewhat spherical structure, which you are finding here, somewhat spherical structure with a prominent nucleus. Now what happens? After some time, there will be the development of a vacuole present in the central area. This is the vacuole. This area is called as the vacuole. Gradually this vacuole will grow in size. It will be full of cell sap, and as a result the nucleus, which was initially placed at the central position, now this nucleus has been pushed here in one corner. Why? Because the central area has now been occupied by the cell sap. Now what will happen? There will be an asymmetric mitotic division. Asymmetric mitotic division in this cell called microspore, and it will result in the formation of one large asymmetric cells, 2 daughter cells. This microspore will divide by mitosis in an asymmetric manner. I said asymmetric, that means there will be one large cell called vegetative cell. So this is vegetative cell, and another smaller cell called generative cell. So at this structure, this structure is called as a pollen grain. So is it clear what is the basic difference between microspore or a pollen grain? Microspore is the structure which is formed as a result of meiosis in the pollen mother cell. This is the pollen mother cell or microspore mother cell. Okay, once again let me use the laser pointer. So this is the pollen mother cell. It has undergone meiosis 1 and meiosis 2. As a result, 4 microspores held together are called as microspore tetrad. This is microspore. Now this microspore will undergo certain changes very soon. Appearance of vacuole, asymmetric mitotic division, and formation of 2 cells. One is called as vegetative cell, one is called as vegetative cell, and another one is called as generative cell. So this is, at this stage, pollen grains are shed. So when the anthers they rupture, this is called dehiscence of the anther lobes, and due to dehiscence or bursting open of the anthers, the pollen grains, the powdery mass of the pollen grains, they will be released mostly under the influence of air current, or they will remain in the flower and they will be taken by pollinating agents to different flowers. So what happens? Exactly when the pollen grain is released, there are 2 conditions. Once again let me use the arrow. So friends, you can see here, this is one pollen grain, okay, and pollen grains are actually yes, when the pollen grain is mature, it contains 2 cells. One is vegetative cell, which we discussed, and another one is called as generative cell. Another one is called as generative cell. So what happens? In over 60% of the angiosperms, the pollen grains are shed at the two celled stage. When the pollen is having, one is vegetative cell and another one is generative cell. In 60% of the cases, pollen grains are released in two celled stage. But what happens in the remaining 40% of this angiosperms, this generative cell, it undergoes division. Okay, it will divide like this, it will divide like this, and it will also have its own nucleus. It will also have its own nucleus here, and it will also have its own nucleus. Okay, and in this way there will be one vegetative cell and two generative cells. Okay, so this is called as three-celled stage. So in 40% of the species, what you will find? In 40% of the species, you will find that the pollen grains are released at three-celled stage. Okay. Now, let us discuss about the structure of a pollen grain. So far as the structure is concerned, it is somewhat spherical. Okay. And you can see that this outermost wall is called exine. Okay. And the innermost wall, this is the inner wall. There, I should say that pollen grain has two wall layers. The outer little darker area is called the exine and the inner one is called intine. At few places, what you will observe, here you can see that the dark black portion of the exine is lacking here. So this portion is called as germ pore. They will help the pollen grains to germinate. Okay. And what will germinate? This generative cell, it will divide into two male gametes and it will come out through the pollen tube through this germ pore. Okay. So it is the generative cell which later on give rise to two male gametes, which we will see later on. What will be the purpose of the vegetative cell? The vegetative cell, it will form the pollen tube. It will form the structure of pollen tube, and the pollen tube will come out through this germ pore. It will come out through this germ pore, and the generative nucleus, it will divide to form two male gametes which will be there in the pollen tube. Now let us discuss something about pollen viability. What is the meaning of pollen viability? You know that the pollen grains, when they mature, the anther wall will be ruptured and they will release pollen grains in the environment. Now the question is, what is the purpose of pollen grain? You will say that the purpose of pollen grain is to pollinate the female sex organ. What is the purpose of pollen grain? The purpose of pollen grain, what is the purpose of pollen grain? The purpose of the pollen grain is to pollinate the female sex organ. It will land somewhere over the stigma and the pollen tube will be formed. But for how long the pollen will remain alive? Well, the pollens, they remain alive, and this life of the pollen grain varies from species to species. Say for example, in cereals like rice and wheat, the pollen grains will remain alive only up to 30 minutes. But there are a number of species. I say Rosaceae, the family Rosaceae or rose family, Leguminaceae or the pulses family, and Solanaceae, that brinjal and tomato and capsicum, all this belongs to Solanaceae family. So the members of these the pollen grains, they will remain viable for several months. And in many cases, the pollen grains are artificially also kept in the preserved condition at liquid nitrogen at -196 degrees centigrade. The pollen grains remain alive. They remain viable. So what is the use of such preservation of the pollen grains at minus 196 degrees centigrade. When we maintain the various pollen grains in alive condition in structures or places, we call pollen banks. In pollen banks, the pollen grains are well preserved and they remain viable. And these pollen grains, they are used by the plant breeders for cross-breeding programs. Okay, so now it's time to see the various beautiful structures. These are the electron microscopic structures of the pollen grains. So this is a very common flower which I have taken for you. This is the well-known China rose, you know. This is called Hibiscus rosa sinensis or Odhul, or sometimes it is also called as Gudhal. So you can see the pollen grain, the structure of the pollen grain here. You might have noticed that when the anthers they rupture, you will find the powdery mass over the flower. It is yellow in color. And if you will observe this under a high power of a microscope, you will observe a structure like this. So this is the pollen grain. You can clearly, these are so many pollen grains here. And what you observe, it has characteristic shape and size and texture. There is a texture over the outermost wall of the pollen grains. These are called as accumulations of the pollen grains or ornamentations of the pollen grains. Because of these ornamentations over the surface of the pollen grains, we can clearly understand it is the characteristic feature of any species. So this is the characteristic feature of the pollen grain. We have the flower called hollyhock in our school garden, and this is the pollen grain of hollyhock. Here in this section we can see the varieties of pollen grains here. Wonderful structures. Although they are microscopic and play important role in fertilization, the pollen grains, the shape, size and outermost wall texture, it is the characteristic feature of different species. So we can, by the help of this unique outermost texture of the wall of the pollen grains, we can identify that this particular pollen grain is of this species. Once again, here I have once again shown a microscopic magnified view. The outermost wall is called exine. Now here I have a special purpose to show the pollen grain once again. This exine is made up of a material, chemical substance called sporopollenin. What is this exine made up of? The chemical nature of exine is called sporopollenin. And what is the chemical nature of intine, the inner layer? The intine is made up of cellulose and pectin. What is the chemical nature of intine? It is made up of cellulose and pectin. And then you can see that this is the vegetative cell and this is the generative cell. This we have already studied. Now, one question is asked on the basis of this exine and sporopollenin. What is the question? Why pollen grains are well preserved as fossils? What does this mean? Friends, you should know that the pollen grains, the outermost wall of the pollen grain is called sporopollenin. It is the most resistant organic material known. Sporopollenin is one of the most resistant organic material known. So no enzyme can degrade sporopollenin, and moreover, because of the sporopollenin, it can withstand a very high temperature and strong acids and alkali.